Okay, so this morning I'm going to talk about analytical ultracentrifugation, which I'll call AUC from now on, and it's one of my favorite techniques. Historically, it's been really important to kind of interpret the huge, big shape and sizes of biomolecules that you can see here in this lovely diagram from David Godsill for the RSCB Molecular Month. Go and have a look at this website. It's interactive, so you might see some of your proteins of interest, but it's also quite nice to kind of get a whole picture of what the different sizes of everything is. And some of these molecules here you might recognize, like the proteasome, and you'll know the proteasome from people talk about it, the 20S and the 26S, or there's the ribosome there in the middle, and you'll know that from people talking about the 50S, 70S, 30S. So all that kind of S is all to do with AUC. And we'll find out later, what that means in terms of the S. But I mean, AUC is not just a kind of historical technique. It's really important for today's science, and I'll show you why. Okay, first of all, I mean, it's a very simple technique. What do? You put a sample in a centrifuge, and you actually spin it, and then you watch it sediment. And you can actually use it for anything. I mean, proteins, peptides, nucleic acids, polymers, polysaccharides, dyes, compound complexes. Why would you want to do it? I mean, there's so many different techniques. Well, I mean, you can ask what's in your sample. You can find out what the mass is. You can find out shape and size. You can look at solvent interactions. You can look at associations, binding affinity, cooperativity, multi-complexes, kinetics, even a binding. And the advantage is it's in solution. There's usually no tags or labels, unless I'll talk about the fluorescence detection system in a minute. And it's a batch measurement, so you've got a fixed concentration. What goes into your sample, that's what you're going to run. So there's no dilution. With the fluorescence technique, it can be a low volume. And the good thing is, hopefully today, when I finish my experiment today, I can recover the samples and we can use them for other things. But you're sitting there, obviously, kind of thinking, Stephen, haven't we talked about a lot of techniques earlier? Wasn't Chris, for those of you who came on Tuesday, kind of going, but it's all these different techniques. With SecMals, assuming surely that could do everything. Well, sometimes you get failures of SecMals. Here's a question, an example here. And as you can see, the squiggly lines there in the middle, that's the masses. And you can see they're very, very sloping across this peaks of separation by size exclusion chromatography. And it means you can't really get a good handle on what the actual exact mass is because it's changing across that peak. And that's because you're going to get dilution down the column. Sometimes it's up to 1 to 10. And sometimes you can't get samples concentrated enough to be able to get species. And certainly in this case, they thought everything was an octamer, and they couldn't really get the concentration up. And it also can be quite difficult to separate the different species. Here, you're relying on separation of the size exclusion, which may not work. Okay, so Chris talked in the single molecule technique about mass photometry. And usually we're in this kind of regime where we're kind of at low concentrations, and you can see nice little point spread functions there, and nice contrast in the center to get the mass. But of course, complexes may not necessarily always form at 20 nanomolar. So if they're kind of higher than that, well, what do you get? You get this kind of sea of massive horribleness where you can't detect really any of the landing species to get any masses. So, so if your complex forms above 100 nanomolar, you can't see it. And also for the instrument that we've got, you can't see anything below 60 kilodaltons. The new instrument, the MP2, can go down much lower. Okay, so for AUC, we can use this for very, very kind of high concentrations and also very, very low concentrations in intermediate as well. And it's also important to can actually deconvolute all these complex samples. So it's an historical technique, but actually it's really kind of been very useful in all these publications recently from the LMB and different projects. And I'll talk about some of those in a minute. Okay, so you think, well, AUC just spins stuff. That's just very simple, isn't it? But it's actually complicated if you actually think about it, because not only have you got to spin something up to like 60,000 RPM, you've also got to be able to film and look at how things are actually sedimenting in real time. And then you've got to analyze that sedimentation. So how do we look at things? Well, you put samples here on the left. This is the cell, and you in this case, in this cell here, you inject the samples into this and through these little ports here into the centerpiece. It's formed and is then got two glass windows clamped onto the top, and then we put it into the rotor, which is then you can actually see all the way through the hole. And the centerpieces we got here is there were 2-sector or there's 6-sector ones. So then light is just shone in the absorbance. Then we're just going to shine light up. It's going to reflect it down through the sample. And then at the bottom, there's a photomultiplier tube. And then there's a radial scanning system. So of course, you got to think this thing is spinning at 50,000 RPM. And this has also got a scan. So how it tells is where the actual sectors are and where the samples are, is there's some magnets in the bottom of the rotor. So it can tell exactly where things are. Huge, big range of absorbance wavelengths we can actually look at. We can scan across the peak. And what we'll then get, you have a reference sample, which is buffer, and then sample in the same reference. And then as everything sediments, you're going to get a change in the absorbance, and that's just purely kind of the intensity change. It's just Beer-Lambert law. You get what the concentration is. So we can also use this interference detection system. So we've got a laser here that's going to fire. It's got 2 slits. It's going to fire through both the sample and the reference. And then we're going to image everything down at the bottom. And that's going to take pictures, one picture of the whole cell rather than having to scan. And so then the camera system, what it does is then it takes then the light beams from the reference and the sample and then combines them all, has a look at it. And of course, what you do is you get interference. And that interference will depend upon the refractive index change between the sample and the reference. So you can get destructive interference, get a black line, or you can get an in-phase kind of constructive interference and get a bright line. And so when you spin a sample, it looks like this. You get this kind of pattern of black and white interference pattern. And how this then curves will depend upon the concentration difference against the wavelength, and that's then related down here at the bottom then to kind of universal constant for the protein to give you what the concentration is. So we can work with what the concentration is across that sector there. So we're quite lucky. We've got one of the few kind of fluorescence detection systems, and this is essentially a confocal microscope setup, and it sits on top sits above the rotor as it spins, and it then scans across. So it's got a solid laser, and then that fires kind of 488 light, which then gets reflected by a dichroic mirror into the sample, and then any fluorescence is then detected by it kind of coming up, goes through the dichroic mirror because it's got a longer wavelength, and then is then focused in through a pinhole here to photomultiplier tube. So it's a really little point here, and then this then scans again as you spin. And it's very, very sensitive, but of course you need a label. But a good thing is that you can actually look at complex samples if you just have one component labeled. So 3 different methods, 3 different detection methods. Which do you choose? Well, absorbance is very sensitive. It's got a limited range, but because we use xenon lamps, there's a peak at 230 where you can have exploit that, and so you can get down a bit further in kind of concentration range. Interference, it's very quick. You've got a big range. One problem, though, of course, is that when you start to get in very high concentrations, then it's difficult to then actually fit. But then in that case, when you're getting very, very high concentrations as well, then you're getting into hydrodynamic non-ideality, which isn't ideal. It's got excellent precision, but one problem, of course, is because it's refractive index change, if your buffers are mismatching between the sample and the reference, then you'll actually see what's happening in the sample and the reference as well. You can see buffer precipitating. Right, so two types of experiment. One, we're going to just sediment very fast. The other one will be in equilibrium. So you usually start off with, with here it goes, and you sediment, and then you watch the scan across during different times, and then you can actually see the concentration change across the radius. So you're going down to the bottom there. So if we then look at it in different time points, then time is coming out of the board, and then the radius is going across here. Then you can kind of see how the concentration actually changes. So we've got to be able to analyze that. And in the next kind of little part, we'll just go through the theory. Okay, we're going to just go through step by step. Don't really worry about the details, but it's really just understanding that it looks very, very simple how to spin things, but actually to analyze it is quite complicated. In the second half, we'll look at velocity equilibrium, and that's much simpler to kind of understand. Okay, so imagine here we've got a sedimenting particle. We've got a centrifugal force that's going to force it down, and then that's going to depend upon the mass of the protein, the angular speed, and obviously the radius as well. But of course, it's not that simple, is it? You've got this particle going through buffer. Like if you're in a crowded, a crowded bar, or you're in a gig or something, and you want to move through the crowd, you've actually got to displace people away to get forward. So in the same way as well, then you've got opposing forces here of friction, and buoyancy. And the buoyancy then is really a measure of how much mass of buffer and solvent you have to get out of the way to actually move that particle down. Also, you're going to have friction as well, and that's going to depend upon the velocity of the particle and this frictional constant here. Okay, but things sediment they're not accelerating down, they're moving at a constant speed down. So therefore, then the sedimentation is really just going to be equal to the balance of these kind of forces here. We just then take out, put in the angular amount, the angular speed, and also here for the buffer as well. And we can just rearrange that nicely to get all the masses on one side. And we're going to substitute this mass of the solvent for the mass of the protein times the partial specific volume of the protein times then the solvent density. And this V bar, we actually saw this on Tuesday. And this is, Chris kind of talked about the kind of the bottom, the change in the effective index, change against concentration for all proteins. And this is from paper from Peter Schuck's lab. And then on the y-axis here, you've got then the partial specific volume of proteins. And you can see it's actually mils per gram. And here's the heat map in red, is most populated. And for most proteins, it tends between 0.2, 0.72 to 0.74. DNA is slightly different. So we can substitute that in, and then we get this nice kind of equation here. So we take out the mass of the solvents. Let's rearrange that. So we've got a constant velocity here. And then we can just rearrange again. So then we're getting then the speed of the particle that's moving relative then to the centrifugal force, or centrifugal speed, sorry. And so this is where this S unit comes up now. This is the Sedberg, and that's what that looks like. And so that's describing the velocity as the particle moves through the centrifugal force. And it's named after the founder of the field, Felix Sedberg. So one S unit is then 1 × 10⁻³ seconds. Now it's important to remember that the S can be 0. It can also then be, it can be 0, or it can be positive or negative. So it can be 0 if you can imagine that, if you see that the S value here that's related upon the solvent density. So imagine if you then increase the density of whatever you're spinning, the medium you're spinning your sample in, then you'll reach a point where basically that will be equal to 1, and then the mass, then that will cancel that top line out, and the S value would be 0. And of course, that's how, when you do density gradients, exactly how it works, because you've got a density gradient, things spin through until effectively all the forces kind of stop everything moving. Okay, so then this frictional coefficient, well, what's this? Well, it's related to the size and the viscosity and really the Stokes radius. and the temperature and the Boltzmann constant. So for BSA, I mean, we've got something that's about, a 4.3 s. So it's moving at this speed at this when it's under this speed of rotor, and it's going to experience a frictional force of 0.05 newtons. So I mean that's a very, very tiny force, but it's still quite important. And if we kind of think about, well, this kind of Stokes radius then relative to a sphere that's of the same mass and density, we can then have this relative frictional coefficient, and that's between 1.2 to 1.5 for globular proteins. So that's a kind of an idea of the shape, and we'll see why that's important later. Right. Okay. So here's the equation. Let's put in. Let's kind of substitute in things to get it in a much neater form. So we can actually then get rid of this. Put in the diffusion here, and then that's going to substitute in. Then okay. Sorry. Yeah, we're going to substitute in, and then we get this equation here, Sedgwick equation. So that's going to be relating then the S value against the temperature, gas constant against the mass, and the diffusional constant as well. So can we solve that equation for everything kind of going through, the kind of going through how things go through? We can think about it in terms of fluxes. So we're getting a concentration change, and so that concentration is moving through down, where we've got a concentration times the speed, and of course the speed is then related to the Svedberg unit relative to the speed of the rotor. And we know that then that sedimentation is going to be then opposed by the diffusion. So that then the kind of the flux at a point is then really going to be a difference of these two. Okay, and so there we're going to put in what the fluxes of the overall pointed are. Let's just substitute in this. And we know that actually the change in concentration is really related to how the flux changes at each kind of point. We substitute that in, and then we get this equation here. And if you look at it, it's a bit of a crazy equation if you look at it. You can't really go any further here. If you kind of look, there's actually concentration over a differential concentration change over radius change on both sides of the equation. So there's not actually any kind of further math you can do to actually solve this equation. So the way they do it is just by doing kind of really simulations, either numerical or analytical kind of methods to try and get through. So essentially what we're looking at then is when things go down, then essentially it's related to the S value, and that's really how fast things are moving down, and also then the diffusion really is how things are going backwards, and so what this kind of slope is. And so then we can analyze everything to get an S value, and that will give us ideas of mass and shape. Right, so for equilibrium, it's a bit easier to kind of understand. Again, if we kind of think about, we're doing relatively slow speeds, and you can imagine it's called equilibrium because everything starts to then kind of stop moving. So that really the sedimentation is then opposed by the diffusion, and nothing's going to move anymore. There's no further movement in the boundary, and so we'll get a static concentration profile like this. So again, when we think about the fluxes, then we've got sedimentation, and then it's opposed by the diffusion backwards. They must equal each other at because of equilibrium. Let's just have a look at again the flux at overall point at R. Then rearrange, and then put in our rearrange again. So then we can then look at the concentration, and then we know where the log is equal to this. Substitute that in again, and then we end up and then integrate. Then you're just going to get an exponential function here. So that's quite easy. So that's related to the diffusion and obviously over below the Svedberg constant number. So let's get rid of the diffusion. If we can then substitute that in, we can rearrange. And now then we've got an equation that's going to relate it then just the concentration that we see against the mass, the angular speed of the rotor against the gas constant and the temperature, and against this is against the radius. So it's quite a simple, then kind of exponential to be able to then solve. So then you can then change. So the exponential steepness is then essentially dependent upon the mass and also how fast you kind of speed things in. Right. Okay. And of course, this it's not, again, you're kind of thinking, well, okay, so far that looks kind of quite easy. But of course, we're not looking at dry samples. And so this buoyant mass is actually, the mass of the molecule and less the mass of the solvent that it's displaced is. But we're assuming it's just that, and there's not like bound water and bound solvent. So then there's actually other terms here, B1 and B3 related to the water and also to any kind of buffer components. The majority of the samples that you use and the buffers that you use, essentially they're kind of quite close in density and viscosity to water. So these kind of terms here kind of disappear, but later on we'll kind of see why they're important. So all that kind of theory, you just have to kind of forget about it now. It doesn't really matter because, again, there's lots of software where you essentially just plug in the constants and then it will actually fit for you. And here's Peter Schuck does analysis of sedimentation velocity experiments, and we can get some of these hydrodynamic kind of parameters just by then the sequences and what the buffer composition is using sedimenterp. We could look at frictional ratio by then actually using modeling, hydrodynamic modeling with HydroPro and SOMO, or we can directly measure things like viscosity and the density with two instruments we've got upstairs. Okay, so how do we do an experiment? Well, I mean, the important thing is, of course, you need pure samples. You need to know what's in your sample. It's good to know what the sequence is. Is there any modifications? You need to know what your buffer components are. So usually, just first of all, put things very, very hard spin, velocity experiment, and do a CNS analysis. If it's not pure, if you think it was pure and it's really looking horrible, then you need to go back and kind of then maybe repurify. Okay, do you want to look at interacting components? Well, yes. Then you can kind of change the distribution of one, or maybe label another one, repeat the sedimentation velocity, or you can do equilibrium experiments and look for the KD. Maybe there's no interacting components. You just want to know, well, can I get the mass? Is that frictional coefficient reasonable or not? And what about then you can do sedimentation equilibrium to find the mass out. Okay, so how much sample do you need? Well, I mean, ideally, if you're looking at absorbance, then you need something that's going to absorb nicely. So you need between 0.1 and 1.3 absorbance units at 280. Do a dilution series if you're looking for interactions. Kind of the volumes, well, for the two sector here, it's 400 microliters, these 12 millimeter sectors. For fluorescence detection, we can go down to 3 millimeter, and you only need about 80 to 90 microliters. Absorbance, then obviously you can do interference or fluorescence detection. And so usually the speed is, in the normal kind of speed of things, then we'll just do things at 50,000 RPM, collect 400 scans. Obviously, if something becomes a lot bigger, then you can slow down the speed, so you can actually then capture how things sediment. And okay, and then you can fit and get the mass and shape, and hopefully some KDs. Now, for equilibrium experiments, this is a bit kind of easier. You just kind of have multiple concentrations over maybe a wide range. You're going to go up and down in KDs. You need all the kind of species to be populated. Smaller volumes, 110 in this 6 sector, 160 in the 2 channel. Again, similar kind of detections. And you do free speed. So one is kind of a slow speed to kind of just for very high mass. And then a middle one, and then you do a very high speed for then just taking everything down to get a baseline. And the thing it does need is because you need to get to equilibrium, you need to get 12 scans, and you need to do that 8 hours. So you need your sample to be able to kind of be happy over like that amount of spinning. And again, the thing we're going to look at is self-association, masses, and equilibrium. Okay, extra considerations. You're going to need to match the buffers if you're looking at interference, absorbance. Yeah, you don't want the buffer components themselves to absorb. Nucleotides, you can look at them by interference, because obviously in absorbance and UV, they're going to absorb a lot. Everything needs to have some salt in there. You need to have at least 10 millimolar in buffer. 120 normal kind of physiological ionic strength is good. Density is near enough as possible to water if you want. Otherwise, you don't want to create density gradients. And certainly if you have glycerol in your sample, then that's not a good idea. Okay, yeah, take out DDT, anything that's going to change over time. And then if you're going to use fluorescence, you obviously need to tag things, but of course, and you need to know the degree of labeling and also remove any free dye. Okay, so let's have a look at some examples that we've kind of used. So here's what your data will actually look like. So on the left, then we've got the meniscus, and then on the right here, we've got the bottom, and then here's the data range. And the color coding here is from start to finish is kind of blue all the way to red. In the kind of the analysis and CS, you just kind of have to put in things like what the V bar is. You calculate that from the sequence. Again, you can calculate from the buffer composition, put in the density and the viscosity. You put in what kind of noise you think might be in there, either time independent or then real independence. Put in, you're going to float the meniscus, and then this is regularization. Okay, so this is an application with Sophia Lövström. And she wanted to know about kind of tau. So she's working on obviously these kind of high tau filaments form. And at an early stage, we think that there's actually a dimeric intermediate. So we went looking for that. And so we took some tau monomer and we spun it. And what we came up with was just one peak that's resolving above the buffer, which is at 0 there. And can we see any dimer? Well, the protein was then sedimenting at 0.9 to 1, and that was consistent with a monomer of about 10 kilodaltons with a very high frictional coefficient. So if you can think that if you have a very high frictional coefficient, it means that something's really extended. So you think that that's going to then have to push through all the buffer and get lots of friction. And so where that will sediment at a much smaller S value compared to S value compared to if it was compact. So it'll sediment like it's a much smaller molecule. So you can see here that the S value then, as the frictional coefficient goes, if you have a massive protein here, if the frictional coefficient goes up, then the sedimentation coefficient goes down. So the kind of kinetics that you can kind of get is that if everything is kind of very, very slow off, then as things sediment, they're going to stay exactly this. They're going to exactly the same. You're not going to get any changes as you sediment all the way down. But if, say, you're forming a complex and the off rate is relatively fast coming off, say 10 to the minus 2, then instead of actually getting two kind of separate peaks of, say, the complex, what you then get is a time average population, because then what you're actually spinning down is you've actually got a reaction boundary spinning down with all the components actually interchanging. But you can actually still fit this, and I'll show you that in a second with this example from David Ronskip. So David Ronskip, they were working on this enzyme here in the endoplasmic reticulum called the FICD, and it's analyzed, and it's got a really weird kind of activity in the fact that it forms a monomer and a dimer, and the monomer is an amylase, but when it dimerizes, it becomes a deamylase. I mean, of course, enzymes can catalyze reactions, and in this case, it's catalyzing in both the directions. And what it does is it analyzes then the molecular chaperone BIP, and so this is going to be then related to how much unfolded protein is in there, and also the ATP levels. So they wanted to understand this dimerization. So when we put it into the AC at different concentrations, what we could actually see, in this case, then what we did was it's in a fluorescence detection system. So I've labeled a small amount of the monomer and then put in lots more of the unlabeled protein. And what we could see is then we weren't getting complex form, but we were just seeing a shift across of the peak. So we could average that individual peaks and get an average weight averaged for each different concentration. And then when we plot that, then you can get a nice kind of change against concentration, and you can fit that to kind of get what the KD for the binding is. They also made a mutant in red, which then showed that when you disrupted that dimer interface, the sedimentation didn't change at all. And here's an example from Paul Elliott. They're interested in then understanding how this ubiquitous was actually formed. So they wanted to understand how Clyde D then was then interacting with Hoyt, and that was important then for deubiquitization of this signal receptor signal complex. And so they find that there was this other protein called SPADA that then seemed to interlink between the two. So we want to understand that complex formation. So we just did simple velocity. And then we could see here in the purple here, which is on your very left there, we've got SPADA, and then blue is the Clyde D. And then when we put them together at different concentrations, we can then see a complex. And as the concentration of the complex increases, you can see that it becomes much more populated and the sedimentation stays exactly the same. From this, then we can actually then detail what the mass and the shape was, and we can work out what the stoichiometry was. Okay, so what do we usually use kind of velocity? Well, questions are like, does my protein self-associate? Do the two components associate? What's the range of species in my sample? What's really in there? Can we get down the mass and stoichiometry? Do I have weaker strong binding? And then, if you've got a multi-component system, what binds to what? So in the question here, this kind of shows you how we can kind of resolve the kind of failure of SAC-MALS. This is a question of, right, how much can my protein dimerize? This is from Jan Lowe's group, this arcadian dimer. And so in SAC-MALS, then the highest concentration, the highest species there was maybe a heptamer, octamer. It was difficult to kind of see. So if we did AUC then at different concentrations and use this analysis here where we know exactly what the monomer is, then we can work out actually how many oligomers are. And you can see at the top there, at highest concentration, there was 8, and then as we start to dilute, then that kind of seems to fall apart. So can we find in a complex kind of binding scenario, can we find out what binds to what? So this was from Bicycle Therapeutics, and they formed these cyclical peptides then interlocked then with a benzene ring there. So you kind of got these two loops, and they then developed this one against TNF alpha, which is a trimeric protein. So they wanted to know exactly where it binds. So we labeled then TNF alpha itself and then sedimented it. We can actually see it kind of forms. There's a trimer, and then there's also a monomer. But there's something in between the two that's not baseline resolved as it should do in between the two. So we think there's a small amount of dimer. Then when we put in just the labeled bicycle and see where that bound, well, the sedimentation there, where it was binding onto the TNF alpha was at about 2. So if we overlay the 2, we can actually see that, yep, it seems to be binding in the middle between the monomer and then the trimer. And actually, when they solved the crystal structure, they actually found out that actually, yeah, the bicycle then forms a cross two then subunits of TNF alpha, which aren't available in the trimeric species. Okay, so we can look at really more complex samples like glycoproteins. We won't go into too much detail, but essentially, like with as Chris was saying on Tuesday, we can exploit differences in the properties between oligosaccharides and proteins. In this case, we're just going to exploit the difference between the Phi bar and the diffusional properties. And so what this paper from Cornwall found was there was an opposing trend of what your theoretical mass would be in black as you increase the amount of carbohydrate on your sample against what the measured sample was. And then essentially you can see where this interaction is between the two. You can kind of get an idea of what the range of percentage of glycosylation is. So I won't go into too much of the detail about that. Now, membrane proteins, they're obviously usually quite difficult to actually look at. And historically, the way people used to look at them was to do density matching, because you've got some usually kind of lipids, or you've got detergent in there to kind of keep them soluble. And so you can imagine trying to, with a protein and a membrane protein that's got lipids or attached, and then you've got in the buffer with lipids, and then you're trying to then match the buffers exactly so you can then completely kind of make the lipids disappear in the analysis. It was quite tricky. Instead, then a new technique here is just forget about trying to density match. Just treat the properties of the detergent and myself, treat them explicitly. And so you can see here in what we've got on the right-hand side here, we've got a peak for the protein. And then on the left there, the smaller is actually the detergent micelle. So we can actually treat that explicitly. The way to do that is, again, don't worry about the details, but we're then looking at the differences in the partially specific volume of the detergent against the protein, because that's what's happening with the spinning particle, and then exploit then the differences in the absorbance between the lipid or the detergent and the protein, and then the differences then in their refractive index changes. So we need to then do measurements with both absorbance and refractive index, and we need to input that in. So we just kind of put that. We put all the constants in, and then we kind of put in what the absorbance is from different detection systems, and then this program then will just kind of calculate what your mass of your protein is, because it'll also work out then what, if we have a look at this lambda d, is essentially down here from just the different signals. It'll work out then what proportion of detergent you've got on there, or lipid, and then you can then work out the plausibility of what the mass is, either from the frictional coefficient change, or from just using the mass of the protein, or from the sedimentation coefficient and the radius. And in this case down here at the bottom, it's really saying, well, is my protein a monomer or a dimer? And essentially, a monomer state will actually fit in what the data actually looks like. Right, here's an application of sedimentation equilibrium. Again, we're going back to this protein, the arcadin, and then we've got 3 different concentrations, and then these are then traces of 3 different speeds, and we fitted this into exponentials, and then this gave us, we used a program here called SEDFAT, give us an average mass of species, which was 116, and then that matched very, very closely then to what the octameric species should have been. Right, so here's a last example then of, again, using sedimentation equilibrium. This is with David Barford's group and Elise Fisher, and they want to look at then the interaction of how the mitotic checkpoint proteins are all assembled. So it's quite complicated. But so they're just honing in on this interaction of MAD, the C-terminal domain, BUB1, and then CDC20. And they wanted to understand how CDC20 was activated and then how that then activates the MAD open to MAD closed. So they know that then, BOB1, kind of the CD1 domain, binds on to the MAD CDT. And then also know that phosphorylation of the MAD1 then promotes the interaction with the CDC20. Okay, so this is all quite complicated. So there must be, in some way then there must be, interactions between the two proteins then with MAD to actually then form this complex. So you can get this mitotic checkpoint complex assembled. So at least she solved the structure of, by crystallography, of the BOB1 and against MAD, the CDT domain. So it's obviously a dimer, it's a homodimer, and so it's binding, the BOB1 here is binding to the coil-coil. And that's what you expect, isn't it? It's a coil-coil, it's, it's binding similar sites. Yeah, that, it looks quite symmetrical, it looks quite nice. Then she looked at the binding constant, and then by ITC, where you just kind of titrate in the peptide into the protein, and the stoichiometry was 1. It should have been 2, because you expect 2 to go in, but it's only 1. That was a bit weird. So then the question is, well, okay, is the crystal structure not right? Is it just a crystal artifact about how things have packed together? So let's do some AUC. MAD alone, yeah, 27. That's that 27 kilodaltons. That look pretty good. Put in BOB1. Oh, it's only gone up to 31, which then means that it's only 1's binding to each dimer of MAD. Okay, well, let's have a look at CDC20. We know that that binds. And if we had CDC20 by itself, that looks all quite nice. And then if we add in MAD1, well, we actually then get only 1 binding there as well. So therefore, the structure is actually asymmetric. So we've got CDC20 binding to one site, and it also binds onto then this globular head as well as the coil-coil. Okay, just a quick summary. Easy to do. Wide range of biomolecules. You can answer this on simple composition, self-association, binding constants, mass, shape, explore mechanism of action. We can tackle really complex samples called membranes, glycoproteins. Okay, lots of literature, lots of books you can read at websites. Obviously Beckman, Peter Schuck. There's also a website, and also in the biophysics interface, that's an internal website. And we're very, very lucky here in the LMB course that we've got dedicated AUC room, and we've got different detection systems and different centrifuges. So come and chat if you want to have any advice, if you want to kind of think about running any AUC. Thanks for coming. Thanks for